Hi, this is the review for Unit 5 Photosynthesis. And the way that I'm going to do each one of these review videos again is that I'm just going to go through each of the learning objectives. So I'll read through each learning objective and then just talk about some important things you should know that relate to each learning objective. And then we will also do some sample problems together. So I'm going to start in Unit 5. Unit 5 is about photosynthesis. And if you want to follow along with me, that is on page 73. This is where all the learning objectives are. So again, this is a really good way to focus your studying, going through each objective here. The other things you're going to want to do is maybe look through old worksheets, look through the PowerPoint, definitely know all the glossary terms, and do as many sample problems as you can. There's 2 practice exams in your lab packet that you can use to test your knowledge. All right, so let's begin at the beginning here. So the first learning objective says, describe the electromagnetic spectrum and the relationship of the parts of this spectrum to one another. So why are we talking about this big thing called electromagnetic spectrum? Well, the reason that this is important is plants need sunlight for photosynthesis, and sunlight is part of the electromagnetic spectrum. The electromagnetic spectrum is just this wide range of types of energy, and all these different types of energy move as a wave, and that's what makes them part of the electromagnetic spectrum. So some examples of electromagnetic energy include gamma rays, X-rays, ultraviolet light, visible light, which is the light that is important for photosynthesis. We also have infrared light, microwaves, or radio waves. What makes each of these different types of energy different from each other is their wavelength. When we're talking about wavelength, we're talking about the distance between the peaks here. So here you can see that microwaves and radio waves have a very long wavelength, whereas things like gamma rays and X-rays have a very small wavelength, the distance between the peaks. One important thing to know about this wavelength is that the wavelength of these different types of energy determines the amount of energy that they have. All right, so things that have a very long wavelength have lower energy. So things like radio waves or microwaves have very little energy, whereas things like gamma rays and X-rays, these have a lot of energy. This kind of makes sense, right? You don't want a lot of gamma rays or X-rays going through your body, or even ultraviolet light. Because of the amount of energy these forms of electromagnetic energy have, they can damage your cells. However, we constantly have radio waves going through our body, and they don't affect our body because they have a very relatively low amount of energy. So why is this important? Again, understanding this wavelength versus energy. Well, each different color of light has a different wavelength, and that's what makes it have a different color. So if we look at white light, like the light coming from the sun, or maybe could be shining a flashlight through this prism here, you can actually separate out all the different colors. So white light is made up of all these different colors of light. And it's all these different colors of light that plants use for photosynthesis. So they can actually absorb this light and absorb the energy in these wavelengths and use it for photosynthesis. If we look at the different colors of light here, you can see that each color of light has a different wavelength. Red light has a very long wavelength, which means that it has a lot less energy than something like violet light. Violet light, the distance between these peaks or valleys is very short, so violet light would have more energy. All right, so that is objective 1. Objective 2 says, describe the method of chromatography and interpret a chromatogram. So before we talk about that, notice again that each of these different types of colors of light have a different wavelength. And plants actually have these molecules called pigments that can absorb different wavelengths of light. And every pigment absorbs a different wavelength or different color of light best. So we can actually see all these different pigments in a plant. And the way that we can see them is by doing what's called a thin layer chromatography. A thin layer chromatography is a method of actually separating out the plant pigments based on their solubility. So the more soluble a pigment is, the further it travels up the strip. So what do I mean by that? See down here, the way you do a thin layer chromatography is that you fill a test tube or some kind of container with this solvent or this clear liquid. This could be something like acetone. And then you're going to stick a piece of paper in here. On the bottom of the paper is a small piece or small drop of plant extract. So you might crush up some spinach and put it on there. As that clear solvent gets sucked up the paper, it's going to carry each pigment up the paper. And notice that these different pigments are separating out. So some pigments can move faster or further up the strip than others. So if you look here, we can see that this orange one moved up really far. This yellow one kind of is in the middle here. Then we have this dark green one and this light green one here. So this is a method that we can use to actually look at how many different pigments are in a plant. And you can separate them out. So the ones that are most soluble travel further up the strip because they're able to be carried or dissolved in the solvent better. So that's why they can travel up further, because it's that clear solvent that's carrying it up. All right, the next objective says calculate RF values from a chromatogram and identify the pigments when given tables of standard values. So from doing this thin layer chromatography, we can then calculate some results from this. And what we calculate as a result of the thin layer chromatography is what's called an RF value or a ratio factor. And the way that you're going to calculate that, you need to know how to calculate this for the exam, for sure, is that what you simply do is follow this formula that's written over here. So this is the formula. Memorize this formula. This is how you calculate the RF. Almost always there will be some sort of RF calculation on the exam. So remember to bring a calculator to your exam so that you can solve one of these problems. So how do we calculate the RF? Well, each pigment will have a different RF. Here we can see that there's 4 pigments. 1, 2, 3, 4. And you can also see where the start is. The start is where the plant extract was first applied. And how you calculate RF is you take the distance the pigment traveled from start and you divide that by the distance the solvent traveled, which was that clear liquid. So the clear liquid's always going to usually travel the furthest. And so you can see that up here. So there's the solvent front, and that's how far the solvent went. Now, one thing to remember is that your RF value is always going to be between 0 and 1. So if you get a really big number or you get a negative number, which you shouldn't, but if you get a number greater than 1, you did something incorrectly. Also remember that the most soluble pigment is the one that traveled the furthest up the strip. So let's do some practice calculating the RF value. So let's calculate it for the orange pigment. If you were to calculate the RF value for this, you would take the distance it traveled from start, which is 5.5 centimeters. You would divide that by the distance the solvent traveled from start, which is 6, and you would get 0.917. On an exam, you will lose a point if you do not calculate it all the way out to a decimal. If you leave it as a fraction, you'll lose a point. So you need to make sure you have a calculator so that you can do that. Notice also there is no units. You're taking centimeters divided by centimeters, so the units cancel out. So RF does not have any units to it. Now go ahead and try to calculate the RF for the yellow pigment, the light green pigment, and the dark green pigment. You can pause this video if you would like to do that. However, here are the answers. The yellow pigment, the RF, would be 3.5 divided by 6, which is 0.58. The light green pigment, you would get 3 divided by 6, which is 0.5. And the dark green pigment, you would get 2 divided by 6, and that's 0.333. So once again, the orange pigment is the most soluble. It traveled the furthest up. And the dark green pigment is the least soluble. A couple of things you have to kind of make sure you don't make certain mistakes on the exam. One is sometimes you'll be given a measurement from the bottom of the paper to start. You want to ignore that because remember, we're starting all of our measurements from the start line. We're not doing anything down here on the bottom. We don't care about the distance from the bottom of the paper to start. Also, sometimes the chromatograms are drawn a little differently than this. So it could be drawn like this. So in this case, if I was asked to calculate the RF for the most soluble pigment, first off, I got to pick which is most soluble. Well, it's going to be the one that traveled the furthest. If I want to calculate the RF for this orange pigment, well, that is going to equal the distance that pigment traveled from start, which would be 3 plus 6. And then I would divide that by the distance the solvent traveled, which would be 3 plus 6 plus 2. And that would equal 9 divided by 11, which equals 0.82. So remember on an exam that you have to show all your work. If you don't show what you divided by what, then you're not going to get credit for a problem like this. All right, so let's move on to objective number 4. It says list the essential requirements for photosynthesis and name 2 important products of photosynthesis. So you should definitely know the equation for photosynthesis. This is extremely important. And this is something that a lot of times students just try to memorize, but think about it logically, okay? What do plants take in and what do plants need? Well, if you think about plants, you know that they give us oxygen, right? So that's going to be one of the products that's going to come out over here. But what do they take in? Well, they take in carbon dioxide. How do plants take in carbon dioxide? Well, they actually take it in through little pores or openings in their leaves. Another thing that plants need, well, think about plants at your house. What do you need to constantly do to your plants so that they can stay alive? Well, you better water them. If you don't water them, then they'll eventually die. So the other thing that plants need is water. How do plants get water? Well, they don't get it through the leaves. They actually suck it up through the roots. Now, the interesting thing happens here. This is really actually very fascinating because plants can build glucose or build carbohydrates just from these 2 starting materials, pieces of starting material. So they can take air, carbon dioxide, and they can turn this into glucose. And if you look at glucose, how does this differ from carbon dioxide? Well, you see that it has a bunch of hydrogens in it. So how plants do that is they actually break down water, they remove the hydrogens from water, and they add it to carbon dioxide. And that's how we get glucose. When they break down water, and they remove the hydrogens, what's left is oxygen. So plants are breaking down water, releasing oxygen, and then they're building up carbon dioxide to make glucose. And glucose is used for energy in plants. It's also used for building up their cell walls. So pretty much plants can build their bodies just from the air and water. And that's just crazy when you think about that. Could you imagine if all you had to do was breathe and drink water and you could grow? So this is basically what's happening here. And all of this is happening in the chloroplast, which are organelles within plant cells that is where light is being absorbed and where this whole conversion photosynthesis process is happening. But one thing that you notice that's extremely important for this process to happen is solar energy or light. So plants have to be able to absorb light in order to make this process happen. So how can we measure the amount of photosynthesis happening in a plant? Well, one thing we could do is measure how much carbon dioxide is being taken in by the plant, or we could also measure how much oxygen is being released by the plant. And that is what you pretty much have looked at in your online lab activities. So one thing that you looked at was this floating disk experiment. And you did this experiment or looked at it in order to understand how photosynthesis is happening. If you notice, these little spinach disks here start to float up after a little bit of time. And what's causing them to float is these little oxygen bubbles on each one of the leaves. So the more oxygen bubbles that are produced, or in other words, the more photosynthesis happening, the quicker these leaves or these disks will rise. So you can actually measure the amount of photosynthesis by measuring how long it takes for those disks to rise. And we called this the FT50. So this was the amount of time in minutes that it took for 50% of the leaf disks to float. So half of the disc, and that's why it has the 50 here. It just means 50% of the discs, how long did it take for them to rise? So one thing that you want to note is that the lower the FT50 is, so the less time it takes for the disc to rise, well, that means the greater the rate of photosynthesis. So how can we calculate a rate from the FT50? Well, the way that you did that in lab was you just did 1 divided by FT50, and that gives you a rate. So that gives you the amount of discs that rise per minute. So that would be the units. So you can easily convert FT50 to a rate. So a lot of times students forget what FT50 means, and then it makes it really difficult to solve some problems. So just when you see this FT50, just think about it as meaning the amount of time it takes for the disc to rise. So let's look at a practice problem here. The following FT50 data were obtained from a floating leaf disc assays conducted under various light conditions, under bright light, dim light, red light, and green light. So this is our FT50 values. So remember, again, FT50, if you read that, just think to yourself, it's telling me how long it took for the disc to rise. So without reading the question, if we just look at the data here, well, we can see that under dim light, it took a long time for the disc to rise. Under bright light, that was the quickest. So the photosynthesis was happening the fastest here because it only took maybe, if this was in minutes, 5 minutes for the disc to rise here. So which of the following is supported by these data? Well, let's go through and read them. Green light is more effective for photosynthesis than red light. Well, is green more effective? Well, no, it took longer under green light, so that's not true. Let's look at letter B. Bright light is least effective. This may be due to heat damage caused by the light. Is bright light least effective? Well, no, that's the most effective. It only took 5 minutes for the disc to rise. C, increased light intensity leads to increased photosynthetic activity. Well, here's where you're changing, the bright and dim is where you're changing the light intensity. And you can see as you increase the intensity, photosynthesis happen at a faster rate. The FT50 is really low. So C looks like it's correct to me. Let's read the last one, just make sure. The leaf discs under dim light have the highest rate of photosynthesis. So if we look under dim light, well, it took forever for the disc to rise. So a higher FT50 means it took longer, so the rate would be the lowest. So we were correct with C. Objective number 5 asks us to interpret the absorption spectrum of a plant pigment and describe the functions of accessory pigments. This is probably one of the most difficult things, I think, from this unit for students to do. So let's quickly talk about how we can interpret an absorption spectrum. So Here's an example absorption spectrum on the right here. And let's talk about how to interpret this. First off, you got to realize that each line represents a different type of pigment. So this is a different type of molecule in a plant that can absorb light, like chlorophyll or carotenoids and so forth. Another thing that you need to recognize here is what these axes mean. The x-axis is always telling you the different colors of light or the different wavelengths of light. And the y-axis is telling you the amount of absorbance. So how much of those different colors of light can be absorbed by that pigment. So remember, each one of these pigments is absorbing a different color of light better than another. Higher peaks equal a higher absorbance, which means that they're able to use more of that light for energy. And that means there's going to be a higher rate of photosynthesis in that color. Wherever you see a low peak, so let's look at this chlorophyll A line. Well, here's a very high peak, so that means it's absorbing a lot of the violet light. But as we go into the blue and green wavelengths of light, you can see that it's not absorbing that light. So if it's not absorbing the light, then what's happening to it? Well, if it's not absorbing it, that means it's reflecting it. And as we talked about in class, that if a certain color of light is not being absorbed by an object and it's being then that means what's happening in this case is it's being reflected. So it's bouncing off of the object. So blue and green light is not being absorbed by this chlorophyll A pigment, and instead it's being reflected. And this is why chlorophyll A is green. Objects appear a certain color because that color of light cannot be absorbed by it and it's just bouncing off of it. So again, if a color of light is not being absorbed, then that means it's being reflected. So if we look at chlorophyll A, part of this objective says to be able to describe the functions of accessory pigments. Chlorophyll A is actually the primary pigment of plants. So this is the most abundant pigment in most plants. This is why plants appear green, because it can absorb violet light the best. However, it reflects green light. It can absorb the green light very well, as you can see in this absorption spectrum. However, most plants don't just have one type of pigment. They're going to have many different pigments in there, a lot of different molecules. And these other molecules are called accessory pigments. Accessory pigments allow photosynthesis to happen over a wider range of wavelengths. If this plant only had chlorophyll A, then it wouldn't be able to photosynthesize at all in this blue-green wavelengths of light. However, by having chlorophyll B, having carotenoids, and having phytoerythrin, these are just some examples of accessory pigments, it allows photosynthesis to happen in colors of light that it typically couldn't happen in if it only had the primary pigment. So these accessory pigments are extremely important in plants. For example, carotenoids, shown in orange here, can absorb like this blue-violet light here and can absorb blue-green light really well. And that's why they can absorb that energy and they can pass that energy on to chlorophyll A. If you notice here with the carotenoids, they're not absorbing yellow, orange, or red light at all. And this is why carotenoids look orange, because they're not able to absorb that light. Instead, they're reflecting it. Carrots have a lot of carotenoids, and that's why they look orange to us. One other point I want to make about absorption spectrums, again, is notice that in most plants, that the least amount of absorbance is happening in the green wavelengths of light. So if you were to look at the amount of photosynthesis happening under different wavelengths of light, photosynthesis tends to be minimal under green light. It's not completely stopped under green light because we do have some accessory pigments that can absorb that light. However, that's where you're going to see the lowest rate of photosynthesis typically. Now let's do some practice problems. These are some difficult problems because now we're going to be applying many of the different concepts we just talked about. All right, so it says the following figure shows the absorption spectrum for a photosynthetic pigment. I recommend for any sample problems that I do is pause the video, try to solve it yourself, and then listen to me talking through it. So here we have a pigment here, and it's showing what light it absorbs. It says, does the color of light absorbed best by this pigment have a relatively high amount of energy or a low amount of energy? Well, where is it absorbing the best? Right here. If I go down, that's a wavelength of about like 460 maybe. Oh no, 460 maybe. So that's like 430 maybe. Well, is that going to be relatively high or relatively low energy? Well, 400 something is a much smaller number than over here. So it means it has a smaller wavelength relative to the other wavelengths in this graph. So remember that the smaller the wavelength, the more energy that something has. So this is going to have a relatively high amount of energy. If this concept is something hard for you to remember, this is what I always think about. I imagine a little person standing here and they're getting hit by waves. If the waves have a very small wavelength, they're going to get hit by these waves really frequently. So it's going to be like they're getting constantly pounded by the waves. So there's a lot of energy hitting them. However, if something has a very long wavelength, well, the waves are going to hit them less often. So there's less energy hitting them. Okay, so that's just something I use to remember that. You could even draw it out to help you out. So this person's getting hit by a lot more energy there with the smaller wavelengths. Let's look at the next question. It says, what wavelength of light will have the highest FT50? All right, don't freak out when you read this word FT50. Just reword it in your mind, okay? So instead, read this as, what wavelength of light will have the highest or take the longest amount of time for it to float? Remember, FT50 means the amount of time it takes for it to float. Well, it's going to take a lot. Which one's going to take the longest amount of time to float? Well, that's going to be where they're absorbing the least amount of light, right? And so that's going to happen somewhere around here, right? So around 500 nanometers. That's how we measure wavelength. So the answer here should be about 500. That's where it would take the longest amount of time for photosynthesis to happen. Let's look at another question here. At what wavelength of light would chlorophyll B produce the greatest amount of glucose? Well, we know glucose is one of the products of photosynthesis, so it's going to be wherever photosynthesis is happening fastest. So chlorophyll B is being shown by the blue line. So let's look here. Well, we can see that this is where our peak is. This is where the most glucose we made. And that looks like it's around there, which is about 480 nanometers. At what wavelength would chlorophyll A have the smallest FT50? So again, reword this. FT50 is the amount of time it takes for it to float. Well, where will it have take the least amount of time for the disk to float? Under what wavelength? Well, we're looking at chlorophyll A now, so the green line. Well, it's going to take the least amount of time for those disks to float where photosynthesis is happening the fastest, right? So that would be where we have the most absorbance. So in this case, it would be around here. That's where our biggest peak is for that one. And it's around 430 nanometers. Hopefully that helps you understand these problems a little better. And just keep practicing with them. The more you practice, of course, the easier they will be. The second and last objective, Objective 6, says identify the following leaf structures and note the functions of each. So you want to be able to identify them and know what they do. So let's go through all these different parts of a leaf. So this is showing a picture of the cross section of a leaf. So imagine I took a leaf and I just kind of cut it with scissors and in a really thin slice, and now I'm looking at it. So this is the top of the leaf and this is the bottom of the leaf here. So what did we see here? Let's focus on the top of the leaf. Well, we see this waxy covering called the cuticle. The cuticle is actually found in the top and the bottom of the leaf. Since it's very waxy, it's not made up of cells. It's actually a wax that the plant secretes. And its main function is to prevent water loss, so to protect the plant. As I said, it's not cellular, so it's not living. Below that, you'll see a single layer of cells called the epidermis. There's an upper epidermis and a lower epidermis. So this is a single layer of cells. It's going to function in protection, and it's also going to help regulate gas exchange and water loss, especially the lower epidermis. Below the epidermis, we have what's called the palisade layer. Notice that these cells are very long and kind of cylindrical, and they are densely packed. So they're all packed together very densely. This is the main site of photosynthesis. So most of photosynthesis happens in the palisade layer, and that is because these cells contain a lot of chloroplasts. And remember, chloroplasts are where photosynthesis happens. So it makes sense that the palisade layer is on the top of the leaf because that's where the sun's going to be hitting it. So that's where it can absorb light the best. If we look at the bottom part of the leaf, we can see the spongy layer. It looks kind of spongy to me, so it's pretty easy to identify. There's a lot of airspace there, so its function mainly is to store gases. Below that, let's talk about how the gases can actually enter the leaves, because remember I said carbon dioxide enters the plant through the leaves and oxygen leaves through the leaves. Well, plants, if you look on their lower epidermis, they have these openings called stoma, or plural stomata. And this is where gas exchange can happen in and out of a leaf. However, plants don't just have these pores or the stoma aren't constantly open. They can actually regulate the opening and closing of the stoma using what are called guard cells, which are shown in pink in my diagram. Guard cells are modified epidermal cells, and they can actually cause that stoma to open or close. Objective 7 actually says describe the structure and function of guard cells. So let's quickly describe how guard cells can open and close. So how do guard cells work? Well, the stoma are open, or guard cells open that pore whenever they fill with water. So when water enters the guard cell, it increases the turgor pressure inside the guard cells, and it actually causes them to buckle out. Okay, so imagine it's getting really filled and it causes it to buckle like that, and then it's open. And so when water enters those guard cells, it opens it up. When will this be beneficial? Well, it's going to allow gas exchange to happen. Plants are going to want to keep this open so that they can bring carbon dioxide in and release oxygen. However, well, then why don't they just keep it open all the time? Well, one bad thing about having the guard cells open, the stomata open, is that water also will evaporate out of the leaves, and the plant doesn't want to lose all this water. So if it's a really hot day, the plant will actually close those stoma so that they can prevent water loss. So how do they reverse this whole process? Well, to close the stoma, water exits the guard cells. And when this happens, so here's my guard cells filled with water. The stoma's in the middle here. Whenever water leaves the guard cells, they become flaccid and they just close the pore or the opening. The benefit of this, again, is it decreases water loss. A disadvantage of it, keeping it closed, though, is that there's no gas exchange. And if there's no gas exchange, they cannot bring in carbon dioxide, which is essential for photosynthesis. So I hope that this helped. I don't think I have any other questions on here. Yep. So if you want to do any more practice problems, look back at your sample problems and look at the practice exam.